Thank you, Tino, and I would like to thank you very much, our host, Professor Claudio Rongo, first of all, for the nice invitation, and the second for this cutting-edge title. And I think I have not to spend so much time being in a meeting dedicated to AKI and with so many experts in the audience and the faculty to revise the principal causes of our AKI. What I would like to do today with you in this talk, just to, first of all, to revise the electrolyte imbalance that are associated with few inherited disease, and then to demonstrate that possibly inherited disease may be one of the causes of acute kidney injury. Let's go to start with the global impact of a rare disease. As you know, rare disease is rare, and so it affects only few individuals in a population. But if we consider the numbers of rare diseases that have been described or individualized, then we end up that roughly about 10% of the total world population is affected by some kind of rare disease. And of this 10% world population, about 50%, as you can imagine, are children, but the other 50% are adults. So this is not only a disease that can be followed up by pediatrician, but also by adult nephrology and the adult doctors. But there are some problems with the rare disease. And the first problem is the definition. You see in Europe, a patient is classified with the rare disease when the frequency is less than 1 every 2,000 individuals. But if we go to the United States, there are a limit number of rare disease patients that has been classified about 200,000. And although in reality, it is presumed that about 30 million Americans are affected by some kind of rare disease. And what is more important is to underline that in order to have a diagnosis of a disease, it takes at least 4.8 years. And with this patient, about 40% of this patient had some diagnosis that was not the right one, as for instance. More importantly, only 5% of these individuals so far they have a drug that is approved by FDA. So the problem is quite important and quite huge, also from the social point of view. There are other things that we should underline, that 80% of rare diseases are of a genetic origin, as you imagine, but there are also rare diseases that may be due to infective, atopic, and degenerative causes. And for the rare disease, the kidney rare disease, we can say that the rare disease is not unusual among patients in end-stage renal disease, because you see that after diabetes, hypertension, glomerulonephritis, and pyelonephritis, the rare disease is the most common cause of end-stage renal disease. As we said, the rare diseases are mostly genetic. They can be monogenic or polygenic. I don't want to go into details. I just want to underline the rare disease is a disease that must be studied. And one step to be studied is that we may have animal models. And for animal models, when we have a monogenic, it's quite feasible. But when we have a polygenic rare disease, then it's a problem. So also to study rare disease at the lab level is not so easy. Let's go now to speak about the rare disease with a major renal phenotype. And if we would like to summarize these issues, we can divide the rare disease in according to the nephron segment. We can have a rare disease that are interested for cells in the glomerular issues, and the predominant feature of this patient is proteinuria and dysmorphic red cell hematuria. Of course, they can have many other symptoms, but that is the prevalent symptoms. If we go now from the glomerulus and we go to the proximal tubule, then we have the disease that some of those are very well known, let's say the proximal renal tuberculosis and the Fanconi syndrome, and the major predominant feature are the loss of protein, low molecular protein in the urine, and the loss of several substances like amino acid, glucose, phosphate bicarbonate, and even calcium. And I want to underline here that so far, at the physiologic, at the molecular level, we know most of the proteins that are located in the proximal tubule, either on the lumina or on the basolateral side, that are implicated in the reabsorption of this substance. These proteins, most of them have been cloned. We know the genes, so theoretically, we are now in the condition to identify most, at molecular level, at the gene level, most of the diseases that affect these cells. If we now move on the thick ascending limb and the early part of this convoluted tubule, we have another group of rare disease, and the predominant feature of this disease are electrolyte disorders, especially affecting the urinary and the concentrating ability. And then you may understand, because actually, you know, the loop of Vera, the early part is the tube, is the so-called diluting segment, very important for the concentration of urine. Moving now to the collecting duct, then we have another group of disorders, and the main feature of this disorder are inappropriate urinary concentration or dilution with loss of body waters, and also the incapability to acidify maximally the urine. And at this level, we know, again, most of the proteins that are expressed. You see, there are some proteins we know quite well. And what is important to underline that these proteins are not only located on the distal tubule or the collecting duct, but are located also in other organs, in particular on the distal colon. On the colon, and you know, sometimes we say that the colon or the GI tract is like a third kidney, and this is very important because when we look at patients that have some disorder of the collecting duct, we also may look at disorder of GI tract, and it's important also because the people that want to study microbiome, they have to consider that the protein that they affect the intestine may affect also the kidney. Well, we have said so far that there are defects expressed especially in the collecting duct and the thick ascending limb, where the salt wasting and the polyuria is one of the main features. So it's possible that a complication of this water derangement and salt derangement may somehow induce acute kidney injury. Let's go to start with to the water imbalance and to electrolyte imbalance in few of these diseases. And I would like to start with disease quite important, that disease that affect the potassium balance. We start with the Bartter syndrome and the Giltman syndrome. These are two diseases that you see, they have some common features. First of all, both patients, they do present hypokalemia. both the two diseases, they have metabolic alkalosis, but there are some features that are different between the Bartter and the Giltman. For example, if we look at the magnesium, in the Bartter syndrome, we have a normal magnesemia, while in the Giltman syndrome, we have a low magnesemia. And if we look at urine excretion of calcium, in the Bartter, we have a large excretion of calcium in the urine, while in the Giltman syndrome, they have a low excretion of calcium in the urine. Why is that? Is that because we have, if we considered that mainly the Bartter syndrome is a disease localized in the thick ascending limb, while the Giltman syndrome is a disease that is localized in distal cuboidal tubule, there are transport proteins that may be not completely affected in the Bartter and not completely affected in the Giltman syndrome. Let's go to look, first of all, to the protein that are involved in the Bartter syndrome. This is cell of the thick ascending limb. And you see that we have a protein on the luminal membrane and protein on the basolateral membrane. And what happens normally? It happens normally that through the sodium-potassium 2-chloride transport system, sodium, chloride, and potassium enter the cells. And eventually, for the potassium, through the so-called ROM-K channels, the potassium recirculates and makes the lumen potential positive. And this is very important, and we express the reason why. And why sodium can exit the cell through the sodium-potassium ATPase, and chloride can exit the cell by the chloride channel. Now, the fact that there is a lumen positive transport potential makes quite easy for the calcium and magnesium to be transported. On the basolateral side, there is another important protein. There is a calcium sensitive receptor, and we know this protein is very sensitive to the calcium concentration in the blood, and may affect by intercellular pathway may affect the function of the sodium-potassium 2-chloride. So if we have a condition in which one of these proteins is somehow affected, what do we have as a consequence? We have the loss of sodium and the loss of chloride and potassium in the urine, and that's we can explain partly of the electrolyte imbalance we have in Bartter syndrome. And secondly, we can have over the loss of calcium in the urine. And this we can explain the increase in calcium excretion. And that is exactly the cases in Bartter syndrome. We have at least 5 different types of Bartter syndrome patient, or type. The first, type 1 is when the sodium-potassium chloride co-transport, the gene is somehow deregulated or changed. The ROM-K is type 2. Then we have the type 3, the chloride channels, and the subunit Bartin is the type 4 of Bartter syndrome. While if we have a gain of function of a gene that encodes the calcium sensitive receptor, then we have the type 5 of Bartter syndrome. But let's go to explain now in more detail why in this patient we have hypokalemia, because this is a crucial step for the Bartter syndrome. First of all, we have to understand that if we have here 2 cells, one of the thick ascending limb cells, and another cell that is the collecting duct cells, you see that the potassium channels, the so-called ROM-K channels, these cells are used just for recycling of the potassium in this segment. While on the collecting duct cells, the potassium channels are used to secrete potassium in the final urine. Now, if you will have now a disease in which the ROM-K, the gene that encodes ROM-K, are somehow affected, we have at the level of the thick ascending limb, we have not the circulation of potassium, and that will make these cells not able to transport potassium. But at the level of the collecting ducts, these cells are in somehow implicated in the secretion of potassium. You may argue that this effect may offset the effect on the fecal assembly limb. But at the level of these cells, we have, in addition to ROMK cells, we have so-called maxi-potassium channels, and these potassium channels are flow-dependent. And somehow they are open when there is a larger flow. And this larger flow will somehow promote the excretion of potassium in the urine. And that's why in Bartter syndrome, the hypokalemia is very well established. And we can have a very dangerous hypokalemia setup. Now, what about the polyuria that is implicated that we have in this patient? Well, polyuria is very well explained because if you somehow along fecal assembly limb you block the capacity to reabsorb ions, then what you do, we have a dissipation of unique concentrating mechanism, and that will explain why in this patient we have also, in addition to loss of potassium and sodium, we have also the loss of water. Now, what about the clinical phenotype? Well, you see, we have several clinical phenotypes. We can have also an antenatal Bartter syndrome. That means these diseases are present during the gestational phase. And but whatever is the phenotype, we have polyuria, hypokalemia, volume depletion, and dehydration. And that can be can very well explain if, in this case, we can have acute kidney injury in these patients. Gitterman. Gitterman, it's a less complex disorder because we have only one gene, at least one gene, and this is encoded for sodium chloride co-transporter, and that's actually will explain most of the electrolyte imbalance that we have in this disease. But speaking about Gitterman, I want to show you some also another disease, with renal phenotype that's quite interesting. And this is called patient affected by the Gordon syndrome. You see, the Gordon syndrome is just a mirror of the Gitterman syndrome. In a patient with the Gordon syndrome are hyperkalemia, while there is hypokalemia in the Gitterman. They do have a metabolic acidosis compared to metabolic alkalosis. They do have hypercalciuria, while in the Gitterman syndrome we have hypocalciuria, and they are hypertensive, while the Gitterman patient, they are at the normal tendency or they have a tendency to have a low blood pressure. So if we have thought that in the Gitterman syndrome, you have an activation, a gain of function of this sodium chloride co-transporter, it was easy to think that in the other disease, we could have just a downregulation of a sodium chloride co-transporter. So geneticists were looking to find if the gene that encoded for this sodium chloride was somehow affected, and they found, unfortunately, no changes in no major damage in the gene encoding the sodium chloride in the Gordon syndrome. And that is quite a nice story. You see that after 35 years from first discussion in 201, Dr. Wilson demonstrated that actually the Gordon syndrome was due to a defect in another interesting family of a protein, this so-called Wnt kinase. This is a very complex issue. I don't have the time. I probably don't have also the expertise to explain all the details, but I want to show you what happens in very simple experiment. This is on oocyte, and you see when you look at oocyte and with the fluorescent microscope, you don't see almost nothing unless you don't inject some protein, fluorescent protein in this oocyte. And then when you inject this fluorescent protein and in the coding for sodium chloride co-transport, you see the sodium chloride transport is well defined on the membrane of this oocyte. This means that the oocyte moves from cytoplasm, the protein moves from cytoplasm on the membrane. But if you co-inject the sodium chloride co-transporter with the wing 4, what you have, that you don't see any sodium chloride on the membrane. This means that this wing 4 can't keep the sodium chloride inside the cells. So it's kind of the pressure to the activity of the sodium chloride. And that is more evident in this experiment done with, for the absorption of sodium in the oocyte. You see, here, this is what the transport rate on the oocyte of the sodium chloride under normal condition. When you inject wing 4, this is much reduced, but what is very, very interesting, when you inject the sodium chloride with a wing 4 that is taken from a patient, you see that this inhibitory effect is gone. So in other words, the wing 4 changes of the genes makes the sodium chloride more effective on the cells, and this, in other words, activate the transporter. Let's go to speak around now about water arrangements. This is taken by Boron Bulp book, and you see here that if we look at the concentration capacity of the tubule, if we go along the proximal tubule, you will have no capacity of the proximal tubule to increase the osmolality of the luminal perfusate. But if we will go now along down to the fin descending limb and up to the thick ascending limb, you see that under condition of water restriction, we have a concentration mechanism and then a dilution mechanism. This is very, very clear. We know from basic physiology. And after you see that there is a concentration becomes more evident to reach a maximum along the collecting duct. Obviously, the concentration mechanism in some account can be disrupted when you have a high water intake. But in these issues, we have to think about that there is another component that is very active. And this component is a AVP, antidiuretic hormone. And you see, if we inject antidiuretic hormone, we can increase the osmolality for the normal 300 millimole up to even 800 millimole. How AVP works on these cells? Well, it works on these cells because in the cells there are some substance or some proteins that are poor, that are the channels, the water channels. The water channels are called aquaporin. They have been discovered not a long time ago. You can find the aquaporin on the luminal side of the cells and also on the basolateral side of the cells. And these aquaporin transport water. Up before their finding, it was supposed the aquaporin, the water flux was mainly paracellular, but now we know that the water flux is a transcellular. And if it's transcellular, we know now that there are several kinds of aquaporin that are present all along the tubules. The one that is inducible is the aquaporin 2 that is expressed mainly on the connecting tubule. And what does AVP? AVP binds to specific receptor, the V2B receptor that are present on the basolateral side, and activate an intracellular cascade, just promoting the formation of cyclic AMP, and this cyclic AMP phosphorylate the aquaporin, so the aquaporin can go from the cells to the luminal membrane, and therefore they are now able to transport via the cells, via cells water. And this is also confirmed in some experiment. This is one very important experiment. You see these dots are aquaporin pure molecules that are in the cytoplasm of convoluted tubular cells. This in absence of VP. When these tubules were perfused with AVP, you see all these dots move on the luminal membrane, and the water permeability of these membranes is increasing. We now know even more, in more and more detail how the AVP works. And this is a cartoon that demonstrated that the aquaporin precursor are synthesized in the rare part of the cells, and then they move from the rare to the Golgi apparatus, and from the Golgi apparatus here, in the presence of AVP, they are phosphorylated, and this phosphorylation moves these molecules on the luminal membrane, and there make their action. In absence of the AVP, what happens? It happens that some of these molecules of the aquaporin 2 are internalized, and some are also degraded by lysosome, and some are excreted in the urine. So you can use aquaporin just to try to find in the urine, and there are some now occasions, some labs that can even make a diagnosis looking at the aquaporin in the urine. Diabetes insipidus. Diabetes insipidus is a very well-known disease that is due to some changes in the production, in the synthesis of ADH. We do have two types of diabetes insipidus, the central one and the nephrogenic tubular diabetes insipidus. In 90% of cases where there is a nephrogenic diabetes insipidus, this is due to loss of a function mutation of the gene that encodes for the receptor or AVP. And this is just an example how this receptor can be affected by changing the genes. But the remaining 10% are related to mutation in aquaporin 2 gene that is present on these chromosomes. And I want to show you a few examples of the importance of this mutation. This is the aquaporin 2. And this is the C-terminal of aquaporin 2. You can have a mutation in different parts. And and I want to show you what happens when we have a mutation in the 256 point. At this site, this mutation, this 526 point is important for an important process, that is the translocation of the precursor of aquaporin 2 from rare to the Golgi apparatus. Or if you have a mutation in this point, what happens? You cannot move further the aquaporin 2 to luminal membrane, and if you now create in the lab a mouse covering the same mutation in the 256 point, what you have this kind of a mouse. This is a control mouse. You look at the kidney, and this is a kidney that carries the mutation. You see that there are the kidney with a very large hydronephrosis. So this is a clear indication of the importance of this point for the aquaporin 2 effect. There is another mutation that we would like to point out is a mutation in 269 point, and here is important because in the absence of this mutation, you cannot have any anchor of the aquaporin 2 on the plasma membrane. So whatever is now the cause of a nephrogenic diabetes insipidus, from the clinical point of view, we have polyuria, we have a polydipsia, and we have nicturia. And in case of extreme fluid loss, the profound dehydration can lead to acute kidney injury. But what about sodium? I would like to come back to this important issue because I want to show you a case, a disease that is related specifically to the arrangement of sodium transport. I just want to remind you that sodium is reabsorbed all along the nephron, Maximally, it's reabsorbed along the proximal tube, where more than 60% of the sodium is transported, but also the fecal sentinel limb takes part of this transport process, and along the fecal sentinel limb, about 25% of the sodium is indeed reabsorbed, and even the distal convoluted tubule, they make a part of this transport process, although at a very low extent. 5% in the distal convoluted tubule, and only 2.5% along the collecting duct. But this the tiny amount of salt, of sodium to transport at this level is quite important, because it is, this segment are after the macrodenser cells. So this means that all the transport of sodium at this level cannot be compensated by the tubulo-glomerular feedback mechanism. At this site, at the site of the collecting ducts, we have one major protein that transports sodium, and this protein is the sodium channel, the so-called ENaC. And the ENaC channels are very important. I want to show you a cartoon where we can define its function. Here we have ENaC on the luminal side. Sodium enters these ENaC channels, and they accumulate in the cells, and they eventually transport on the basolateral side by sodium-potassium ATPase. But the accumulation of sodium into the cell by the ENaC, what does? Make the lumen more electronegative in this instance. And being the lumen more electronegative, what you have that the secretion of potassium is favored through the ROMK channel, and this also favor the secretion of a proton into the lumen. So reabsorption of sodium through the ENaC cells, the ENaC channels, promote the secretion of potassium and promote the secretion of hydrogen ion. So in other words, and so if you have now a disease where you have activation of these ENaC channels, what you have? Loss of potassium and the loss of hydrogen ion. In other words, you can have metabolic allosis on one side and hypokalemia on the other side. And this disease is exactly the Liddle syndrome. Liddle syndrome, you see, is quite a rare disease in which the clinical signs, the most important clinical signs, are hypertension and metabolic alkalosis and hypokalemia. And this disease, the plasma blood aldosterone is suppressed, because if you transport more sodium independent from aldosterone, you suppress aldosterone and you don't activate this hormone. So hypertension on one side and the hypokalemia on the other side are the major issues of Liddle syndrome. And here is a cartoon. What happens in this patient? You have changes in XY motif of the channels, and the channels are always in the open state. If in the open state, sodium may enter the cells easily, and entering the cells, obviously it can be transported. And that's one example, one experiment done on animals with the Liddle disease, compared to animals of wild type. These are collecting duct cells, and you see here that the sodium channels are all present in the cytoplasm of the cells. When you shift these animals to a normal sodium diet, to a lower sodium diet, what happens? You happen that all the channels from the cytoplasm go on the luminal membrane, and that's what physiologically you expected. And when you the animals from a low sodium diet go back to a normal sodium diet, you see that all the channels from the luminal membrane, they are going on the cytoplasm. Now, if you have a mouse with a mutation of the sodium channels, exactly as it is in the patient with Liddle syndrome, you see that the sodium channels are all in the cytoplasm when they are fed with the normal diet. Low sodium diet, they go on the plasma membrane, but when the diet is changed to normal, they stay on the plasma membrane. So there are no way that it can be recruited in the cytoplasma. So to study this rare disease, it's important for, let's say, for the patients affected by the disease. But also I think it's important for other patients. And I want you to give an example of this. We have studied the Liddle syndrome, and we have defined that Liddle syndrome is due to activation of a sodium channel and is characterized by hypokalemia. Now, the obesity-related hypertension, that's not quite rare, has features that are similar to Liddle disease. In fact, this hypertension is characterized by hypokalemia. Sometimes it's resistant to conventional therapy, and it may be due to the accumulation of sodium channels. In other words, to study sodium, the rare disease is important also for patients that are not affected by this type of disease. Let me finish with a couple of slides, just mentioning what happens now in Europe. The European community has somehow recognized the importance to study rare disease. And now they have launched a program, the so-called European Reference Networks. In the European Reference Network, there are thematic areas. And going from rare skin disorder to cardiac disease. And you see that is present also the disease that have expression on the renal phenotype. And they have been already called. They are there that you see all the centers that have been recognized. Italy is quite well represented, but there is another call at the end of this year, so more centers can participate to these European efforts. But in order to do study on the rare disease, we must be able in our lab to identify very well the phenotype. And in particular, if we want to study from the tubular disease, we must have in our hands the possibility to perform functional tests, just to look at the proximal tubule, to look at the ficus and the limb, and to look at the distal tubular cells. But we must also be able to measure the glomerular filtration rate, because to use the so-called formula to identify glomerular filtration rate in this patient is, I think it is not affordable. It's completely not affordable because we do have different process along the tubules. But obviously, we must be also able to do genetic analysis. And not only genetic analysis using next generation sequencing, but also to test next generation sequence to a panel that will allow us to identify, to sequence the Mendelioma, and in the future also to look at the whole exome and the whole genome. I think with all these assets in our hands, we will be able to identify nowadays most of the renal rare disease. We can also understand what are the electrolyte imbalance in disease, and possibly we can add to acute kidney injuries another cause of disease. And before to finish, I would like to thank all of you and Professor Ronco for the kind invitation, but I want also to thank Cristina Masella, this young fellow of mine that's helped me to prepare all these slides and also this talk. Thank you very much.